So in class today we talked about replication of DNA, and what we're going to talk about tonight is how to make protein. So before we made more DNA so that the cells could split. Now we're making protein that goes to work and does its functions throughout the body, building things and whatnot. So we're about to see how our body makes proteins. So it's going to start out a lot like what we talked about today. Okay, we're going to start out with a DNA molecule. Okay, so it's going to be like this. Okay, you guys have seen me draw this before. Okay, so it's going to look a little bit like that. A double helix, remember, it's got two sides, okay? And we're going to uncoil it, okay? Helicase, remember helicase is going to uncoil the DNA, and then it's going to look like a ladder. Okay, and you remember that there are nucleotides on either side, okay? It's a little bit harder to write on this than it is to write on the board. Okay. Alright, so helicase has uncoiled our DNA just like what happened in our DNA replication, okay? So what's going to happen next is it's going to unzip, okay, just like it did in the other process, okay? So we've got some that are still connected, okay, because it's unzipping from the bottom, okay, and it unzips from the bottom up, okay? So we've got the A, T, C, G, G, C, C, G. Except for these are starting to break apart. Okay, these hydrogen bonds that are between them are starting to break. Okay, open up a new page. Alright, so we are going to go from this point, okay, where the DNA molecule has started to split down the middle. The hydrogen bonds between the nucleotides have started to break. So now we look like this. Okay. This is a little different because the DNA is not going to completely unwind all at once. Okay. But it's the same deal where we've got the nucleotides here. Okay. We got them on this side too, although I'm only going to draw them on the other side because this is taking a while. So what we're going to do is we're going to make some mRNA. M stands for messenger, okay? And these are a bunch of RNA nucleotides. Remember I told you that in RNA that A binds with U instead of with T. U stands for uracil. So uracil instead of thymine. Okay, so we've got all these nucleotides floating around out here. Okay, and RNA polymerase instead of DNA polymerase is going to come and pick these guys up and take them to match up with this template. Okay, so it's going to take a G and it's going to bind right here. Okay. Okay, and then it's going to take a T, or it's going to take a U because we don't have T's, and that's going to bind right here. Okay, and we're going to make a strand of RNA. That strand of RNA, it's called a strand of mRNA, messenger RNA. That's because that RNA is going to take a message, this code, to the ribosome. So it's going to make this whole strand, okay, it's going to finish off the whole strand, and it's going to look something like this. Okay, it's going to look like this. U, G, C, G, U, C. Okay? So this is the complementary strand to this one. Okay, it bound here, and then it was released, okay? And this mRNA is going to go to the ribosome. Remember, there are ribosomes in our cells, okay? So this is all happening in the nucleus, but now this mRNA is going to leave the nucleus and go to the ribosome. That's where we're going to finish making the protein. Okay, so now we've got this mRNA. I'm going to draw a ribosome for you, okay? It's going to look kind of like a spaceship, but it's a ribosome. Alright, and the mRNA has just come from the nucleus, okay? And it's got the nucleotides on it, just like we said before. U, G, C, G, U, C. I'm going to add one at the end here, A. Okay? So this is what we got from our DNA template before. This is the mRNA that we got from the template. Alright, and it is going to travel through this ribosome, and as it goes through the ribosome, there are this other form of RNA. It's called tRNA, transport RNA, and it looks kind of weird. It looks kind of like a pitchfork. Looks like that, and it's got an amino acid on the end. The amino acids are what build proteins. Okay, so there are these tRNAs with amino acids on the end. Okay, tRNA with amino acid on the end. Okay, and these all have codes that match up to these guys. Okay, these are going to match up just like we matched up with the template before. So this one, these are going to be translated in groups of 3. 1, 2, 3. 1, 2, 3, just like that. Okay, so UCA matches up with A, G, U, okay, and then GCG matches up with CGC. Okay, so this is the complementary tRNA to this one, and this is the complementary tRNA to this one, and these are each carrying an amino acid. Alright, I'm going to go to the next page. So the mRNA is going to go all the way into the ribosome like this, okay, and now our code is on the inside there. 1, 2, 3, 1, 2, 3. So I'll just do the ones that we were talking about a second ago. A, C, U, G, C, G. Okay, so this is still our mRNA. mRNA. Okay, our tRNAs are floating around all out here, okay, and they're going to recognize that they can match up with these guys. So that tRNA that can match up right here is going to come and bind and bring its amino acid with it. Okay, so this one had an A here, and a G here, and a U here. Okay. The tRNA that matches up with this codon, this group of 3 mRNA, okay, is going to come and bind. This is a C and a G and a C. These guys bind together, okay, and it's got an amino acid here. Now, our whole point is to make a protein, which is a chain of amino acids. So now that these are lined up in the right order, these guys can form a peptide bond. Alright, and that is going to continue all the way down the chain with all of these. So now it would move on to this next codon and just shift out, shift in this direction as it goes. Okay, and make a long chain of amino acids. Alright, that's called a protein. So that's what we're going to continue to go over tomorrow. We're also going to go over some mutations tomorrow. So take good notes on this, draw some pictures, and I'll have a picture for you guys as well later on another day. Okay, so go ahead and make sure you write down the pictures you can, grasp it as well as you can. This is also in your Power Notes. Okay, so look at your power notes some too. Alright, I'll see you guys tomorrow.